All right, hey everyone, welcome back to the deep dive. I was thinking about something pretty wild the other day.
Oh, do tell. Always up for a wild thought.
Well, you know how we all start as like this single itty-bitty cell, right? A fertilized egg.
Yeah, the very beginning.
Right. And now, here we are, all grown up with trillions of cells. And get this, they're all so different. You've got your nerve cells zapping signals super fast, muscle cells flexing and contracting, skin cells tough as nails, protecting us from, well, everything.
A symphony of cells, each with its own unique role to play.
Exactly. But here's the thing that really blew my mind. Every single one of these cells, no matter how different they seem, has the same exact instruction manual, the same genome.
Ah, yes, the paradox of cellular diversity. How can such a diverse array of specialized cells arise from a single, unchanging blueprint?
That's the million-dollar question, and I bet it's a question some of you out there, especially Mr. Martin's IB biology students, have pondered as well. So how do we go from one generic cell to this mind-boggling diversity of cell types, all using the same DNA?
The answer lies in the fascinating world of gene expression. Think of your genome, all your DNA, as a massive cookbook filled with countless recipes.
Okay, I like where this is going, a big foodie.
Gene expression is kind of like choosing to read and follow a specific recipe from that cookbook. The recipe, in this case, is a gene, a segment of DNA that holds instructions for building something, a protein.
So proteins are like the dishes we're making.
Exactly. Proteins carry out all sorts of crucial tasks inside the cell and ultimately determine the cell's characteristics, its phenotype.
It's like how different recipes lead to different meals. Got it.
That's why your eye cells look and function differently from your liver cells. They're each expressing, or reading and following, a distinct set of genes from the same cookbook.
Okay, that makes sense. But it can't be completely random, right? I mean, cells
Yeah.
don't just flip through the cookbook and pick recipes willy-nilly.
Oh, absolutely not. There's a lot more to it than just reading. It's about carefully controlling which genes get read, when they're read, and how much they're read. It's like having a master chef in the kitchen who decides exactly which recipes are needed and when.
So there's regulation, like a control system.
Precisely. Gene expression involves two main stages. First, we have transcription, where we create a temporary working copy of a gene's instructions. In biological terms, this means making a messenger RNA or mRNA molecule from a DNA template. Think of it like jotting down the essential steps from the recipe onto a separate card.
Like a cheat sheet so you don't have to keep opening the giant cookbook.
Exactly. And then comes translation, where that mRNA cheat sheet is used as a blueprint to actually build the protein. This is where the cooking happens, turning those instructions into a functional molecule.
Okay, so we've got the cookbook, DNA, the recipe card, mRNA, and finally the dish, the actual protein. But you mentioned control and efficiency. Why is that so important?
Cells are remarkably efficient. They don't want to waste precious energy and resources producing proteins they don't currently need. That's why regulation is so critical.
So it's not just about the how of gene expression, but also the when and the how much. It's like knowing exactly how many servings of each dish to prepare based on the number of guests you're expecting.
Absolutely. And that's what we'll be exploring in this deep dive, how this intricate regulation of gene expression leads to the incredible diversity of cells and their specialized functions within your body.
This is already getting pretty complex, but it's totally fascinating. Now, for anyone tuning in, and I know we've got some international listeners and students, perhaps even those in Mr. Martin's class who are learning English as an additional language, don't worry if you get tripped up by some of the scientific jargon. We'll try to make things as clear as possible, and you can always rewind or look up any terms you're not familiar with.
We'll be using plenty of everyday examples and analogies so no one gets left behind.
Great. Now let's get down to the nuts and bolts. How does a cell actually decide which genes to read in the first place? How does it know what to do?
It all begins with transcription. Remember that first step. The control happens right there. Not every gene is active all the time. Some are turned on, some are turned off, and some are set to low or high depending on what the cell needs at that particular moment.
So it's like a control panel with tons of switches for each gene. On, off, dimmer switches.
Exactly. And two key players in this control panel are promoters and transcription factors. Promoters are like road signs, specific DNA sequences that sit right next to the start of a gene. They act like designated parking spots for RNA polymerase, the enzyme that does the heavy lifting of reading DNA and making the mRNA copy.
So the RNA polymerase needs to know exactly where to start reading. It needs a clear landmark.
Precisely, and that's where the promoters come in. Now, transcription factors are like the actual switches on the control panel. They are proteins that bind to these promoter regions, either helping or hindering RNA polymerase from doing its job.
Okay, so the promoters tell RNA polymerase, park here, and the transcription factors act as the on or off switches. But you said helping or hindering. It's not just a binary on or off, right?
Right. Some transcription factors are like boosters, encouraging RNA polymerase to get to work and produce more mRNA. These are called enhancers. Others, called silencers, do the opposite. They block RNA polymerase from binding, essentially silencing the gene.
So we've got enhancers that ramp up gene expression and silencers that put on the brakes.
Exactly. It's like a volume knob, with different transcription factors fine-tuning the level of mRNA that gets produced from a particular gene. And to make things even more interesting, there are also enhancer regions in the DNA where activator proteins can bind to really boost transcription.
So it's like having a super boost button to crank up the volume even further.
Right. And there are also silencer regions where repressor proteins can bind to really shut things down tight. It's this whole intricate interplay of multiple factors working together to fine-tune the levels of gene expression.
Wow, so it's not just on or off. It's this dynamic system with enhancers, silencers, activator proteins, and repressor proteins, all working together to precisely regulate how much of each protein gets made.
It's quite a symphony, really, and the regulation doesn't end there. Even after an mRNA copy is made, the cell has other tricks up its sleeve to control protein production.
Tricks. You're making it sound sneaky.
Well, perhaps strategic is a better word. This brings us to the control of mRNA degradation.
Degradation. That sounds a bit like the cell is intentionally sabotaging its hard work.
In a way it is, but it's all part of maintaining balance and efficiency. Remember those mRNA transcripts, those recipe cards? They aren't designed to last forever. The cell can actually control how long they stick around.
So it's like setting an expiration date on the recipe card.
Exactly. By breaking down mRNA molecules, the cell limits the amount of protein that can be made from them. And the lifespan of an mRNA molecule can vary quite a lot. Some only last a few minutes, while others can persist for days.
So a short-lived mRNA will only lead to a small burst of protein synthesis before it's gone.
Precisely. Now, mRNA molecules do have some built-in defenses to protect them from immediate destruction. At one end, there's a structure called a guanine cap, and at the other end, a poly-A tail, which is a long string of adenine bases.
Like protective armor on the recipe card.
Exactly. Think of them like those little plastic tips on the ends of shoelaces that prevent them from fraying. They help protect the mRNA from being immediately chopped up by enzymes.
So these caps buy the mRNA some time, but eventually they do get broken down.
Yes, eventually. There are enzymes called exonucleases that specialize in dismantling mRNA, starting from the ends.
So they're like tiny shredders, chomping away at the mRNA from the edges.
A perfect analogy. But here's the catch. They can't easily attack the mRNA while the cap and tail are still in place. First, other enzyme complexes need to step in and remove those protective ends.
So it's a two-step process. First, remove the armor, then shred.
Right. The decapping complex removes the guanine cap, and the dedenylase complex shortens the poly-A tail. Once those protective barriers are gone, the exonucleases can move in and break down the mRNA molecule. And what's really interesting is that the length of the poly-A tail can actually serve as a signal.
A signal for what?
A shorter poly-A tail essentially marks the mRNA as more vulnerable to these degradation enzymes. It's like putting a big red X on the recipe card, signaling to the shredders that it's time to get to work.
So the longer the tail, the more protected the mRNA and the more protein it can make. But if the cell needs to slow down protein production, it can shorten that tail, making the mRNA more susceptible to degradation.
Exactly. The cell can actually control the length of the poly-A tail. And even hormones, those chemical messengers that travel throughout the body, can influence this length, adding yet another layer of control over protein production.
So we've got this amazing system where the cell can regulate gene expression at multiple levels. It can control when and how much mRNA gets made, and then it can also control how long that mRNA sticks around before being degraded. It's a really fine-tuned system.
A true marvel of cellular efficiency, wouldn't you say?
Absolutely. Now let's go back to our original question. How do we get this amazing diversity of cell types all starting from the same DNA blueprint?
That brings us to epigenesis.
Epigenesis. Sounds complicated.
It's a big word, but the concept is actually quite straightforward. It's the process by which cells in a multicellular organism like us become specialized. They take on different roles, different identities. Think back to our examples:
Hmm.
nerve cells, muscle cells, skin cells, all those specialized types we talked about.
All this specialization happens even though they all have the same DNA, the same genome.
Hmm.
That's incredible.
That's the beauty of it. The same instruction manual, used in countless different ways. Different cell types simply express different sets of genes. For example, a liver cell has all the genetic information to be a nerve cell, but it doesn't express those nerve cell genes. It selectively activates only the genes it needs to perform its specific job as a liver cell.
So it's not about changing the cookbook, it's about choosing which recipes to actually cook.
Precisely. Each cell type focuses on a specific chapter, a subset of genes that are relevant to its function, and ignores the rest. And this selective gene expression is what leads to the different phenotypes we observe the appearance and behavior of cells without any alteration to the underlying genotype, the DNA sequence itself.
So the DNA is like the potential, and gene expression is about realizing that potential in specific ways.
A brilliant way to put it. Think of stem cells, those remarkable cells that have the potential to become any cell type in the body. When a stem cell decides to become a nerve cell, it starts ignoring all the other recipes and focuses solely on the instructions for being a nerve cell. It turns on nerve-specific genes and keeps the rest silent.
This is really mind-blowing. So to understand a cell, we need to look beyond just its DNA sequence. We need to understand which genes are being expressed, which recipes are being followed. And that brings us to these three key terms we hear a lot in biology: genome, transcriptome, and proteome. Can you break those down for us?
Of course. We've already talked about the genome, the complete set of genetic information in a cell. All the DNA, the full cookbook. With very few exceptions, like mature red blood cells, every cell in an organism has the same genome. Now, the transcriptome is the entire collection of RNA molecules in a cell at a given time. This includes all the mRNA transcripts, those recipe cards we talked about.
So it's like a snapshot of which recipes are currently being copied and used in the kitchen at that moment.
Exactly. And the transcriptome can vary greatly between different cell types. A nerve cell's transcriptome will be very different from a muscle cell's transcriptome because they need different proteins to carry out their unique functions.
Okay, so genome is the entire library, and transcriptome is like the pile of books that are currently checked out and being read. What about proteome?
The proteome is the entire set of proteins that a cell is producing at a given time.
So the proteome is the actual meal, all the dishes being prepared based on those copied recipes.
Precisely. The proteome is even more complex than the transcriptome because sometimes a single mRNA molecule can actually be translated into multiple, slightly different protein versions. And just like the transcriptome, the proteome also changes over time depending on the cell's needs and its environment.
So we've got the full library of recipes, genome, the recipes currently being copied and used, transcriptome, and the final dishes being made, proteome. That helps paint a really clear picture. Now, earlier we talked about how this regulation, this control over gene expression, is achieved. You mentioned something called epigenetic tags. What exactly are these tags and how do they work?
Imagine epigenetic tags as sticky notes attached to your DNA. They don't change the underlying DNA sequence itself, but they can signal to the cell whether a particular gene should be read or ignored. They can say, read this one or skip this one.
Okay, so they're like little notes that say important recipe or don't bother with this one.
A great analogy, and one of the most common epigenetic tags is DNA methylation.
Methylation. Doesn't that involve adding a methyl group, which is the CH3 molecule, to the DNA?
You're exactly right. In DNA methylation, a methyl group gets attached to a specific DNA nucleotide, often to a cytosine base, especially when it's located in the promoter region of a gene.
Remember, the promoter is that park here sign for RNA polymerase. So if a promoter gets methylated, what happens?
Well, often methylation acts like a do not enter sign. It makes it much harder for RNA polymerase to bind to the DNA and start transcribing that gene. It essentially silences the gene.
So methylation is like putting a big X over that recipe, telling the cell, don't bother with this one.
Precisely. Now, it's not just about blocking the starting point. Methylation can also happen on histone proteins, those proteins that DNA wraps around to form structures called nucleosomes.
So DNA is like thread and histones are like spools that help keep it organized.
A helpful image. Adding methyl groups to histones can either repress or activate transcription, depending on exactly where on the histone the methylation occurs. For example, methylation of a particular amino acid, lysine, at position 9 or 27 on histone H3 tends to stop transcription.
Why does that happen?
Well, in this case, methylation can make the DNA wind more tightly around the histones, making those genes less accessible for transcription. It's like packing those recipes away in a box at the back of the cupboard. On the other hand, methylation of lysine at position 4 on H3 can loosen the DNA structure, making it easier to transcribe those genes. It's like putting those recipes right on the counter, ready to be used.
Wow. So it's not just about turning genes on or off. It's also about how accessible they are, how tightly packed they are, and methylation plays a big role in all of this.
Absolutely. And here's where things get even more interesting. Those epigenetic tags, those sticky notes on your DNA, can sometimes be passed on from one cell to another. It's called epigenetic inheritance.
Inheritance, you mean like from parents to their children?
Yes, sometimes even across generations. Epigenetic tags can be copied when a cell divides, so the daughter cells inherit the same methylation patterns as the parent cell.
So it's like copying those sticky notes onto the new recipe books for the next generation of chefs.
A good way to think about it. And even more fascinatingly, some epigenetic tags can actually be passed from parents to their offspring through sperm and egg cells.
So experiences a parent have could potentially affect how their child's genes are expressed.
That's the current thinking, and it's a hot topic in research right now. This means that traits can be inherited without any changes to the actual DNA sequence itself. It's all about which genes are turned on or off.
That has huge implications for our understanding of heredity. It's not just about the genes themselves, but also about how they're regulated.
Exactly. Now, it's true that during the formation of sperm and egg cells, many of those epigenetic tags are removed in a process called reprogramming. It's like wiping the slate clean for the next generation. But some tags manage to slip through the cracks, and those are the ones that can be passed down to offspring.
So it's not a complete reset. Some of those parental notes, those epigenetic tags, remain.
Right. And those persistent tags can have fascinating consequences. For example, think about cell specialization, how cells become different types. A nerve cell, when it divides, will pass on not just its DNA, but also its specific pattern of methylation, ensuring that its daughter cells also become nerve cells.
It's like passing down a tradition, a cellular lineage.
A great analogy. Now, we briefly mentioned earlier that the environment can play a role in all of this. How do external factors, things outside of the cell, actually influence gene expression through these epigenetic mechanisms?
Yeah, that's what I was wondering. What kind of environmental cues can rewrite those sticky notes on our DNA?
Well, a really striking example is air pollution. Studies have shown that exposure to air pollutants can actually lead to changes in DNA methylation patterns. Specifically, it can often lead to a decrease in methylation in certain genes.
And remember, methylation often acts as a silencer. So less methylation means those genes become more active.
Exactly. And this can have a range of effects, both good and bad. Interestingly, many of the genes that are affected by air pollution-related demethylation are involved in the immune system.
So our body's defense system is directly impacted by the air we breathe.
It appears so. On the one hand, increased expression of immune genes could help the body fight off harmful substances in the polluted air, but on the other hand, it can also contribute to health problems like asthma and heart disease. There's also evidence that air pollution can affect methylation patterns in genes related to pregnancy, potentially leading to adverse outcomes.
So it's a double-edged sword. The environment can both trigger our defenses and make us more vulnerable at the same time. It's like the air we breathe is literally rewriting the instructions in our cells.
It's a powerful reminder that our environment and how we interact with it can directly impact our biology at a very fundamental level, right down to how our genes are expressed.
Now, you mentioned earlier that not all epigenetic tags are wiped clean during the formation of sperm and egg cells. What are some of the interesting consequences of those tags that manage to persist across generations? Can you give us some real-world examples of how this plays out?
One particularly fascinating example involves ligers and tigons. You've probably heard of them.
Oh yeah, ligers are those huge, majestic creatures that are a mix of a lion and a tiger. Yeah.
And tigons are the offspring of a male tiger and a female lion. Now, here's where epigenetics comes in. Male lions have certain genes that promote the growth of their cubs.
So lion dads want their cubs to be big and strong.
Exactly. It's thought that this evolved because female lions often mate with multiple males, and so there's competition among the cubs. The dad's genes are basically trying to give his cubs a size advantage.
Makes sense, survival of the fittest and all that.
Right. And it turns out female lions don't have these same growth-promoting genes. Now, tigers are different. Female tigers usually mate with just one male, so there's less need for these super strong growth genes.
So lion dads are all about make my cubs huge, while tiger dads are more chill about it.
You could say that. Now, when you cross a male lion with those strong growth-promoting genes with a female tiger who lacks those genes, you get a liger. And guess what?
A supersized cat.
Exactly. Ligers often grow to be much larger than either lions or tigers. Those growth genes from the lion dad are firing on all cylinders, and there's nothing from the tiger mom to counteract them.
So it's like the lion dad's instructions are saying grow big, grow fast, and the tiger mom's instructions are just silent on the matter.
Precisely. Now, with tigons, it's the opposite. The male tiger doesn't have those strong growth genes, and the female lion might still have some leftover tags that inhibit growth. So tigons are often smaller than ligers and even smaller than their parent species.
Wow, so these epigenetic tags, these inherited notes from the parents, can lead to really dramatic differences in size.
And that's just one example. Epigenetic inheritance is likely playing a role in many other traits and health outcomes as well. It's a whole new layer of complexity that we're just beginning to understand.
It's really changing how we think about heredity. It's not just about the genes themselves, but about these inherited patterns of gene expression.
Precisely. And to really study the interplay between genes and the environment, scientists often turn to identical twins. Have you ever wondered why?
Well, I know identical twins have basically the same DNA, right?
They do. Identical or monozygotic twins come from a single fertilized egg that splits, so they start with virtually identical genomes. This makes them perfect for studying how the environment can affect gene expression. Because if you have two individuals with the same DNA, but they grow up in different environments or have different experiences, any differences you see between them are likely due to environmental factors.
So it's like a natural experiment.
Exactly. And one of the ways researchers string this is by looking at methylation patterns in identical twins. They found that early in life, identical twins have very similar methylation patterns. But as they get older, those patterns start to diverge, especially if they live in different environments or have different lifestyles.
So even though they started with the same blueprint, their life experiences are leaving different epigenetic marks on their DNA, leading to different patterns of gene expression.
That's right. And our source material doesn't go into detail about this particular study, but I think it's worth mentioning that there's been a lot of interest in studying twins, where one has experienced a dramatically different environment, like one twin going into space while the other stays on Earth.
Wow. That would be an extreme example of environmental influence.
Right. And even twins raised in the same household might not experience the same subtle environmental cues or have the exact same health issues, which can lead to different methylation patterns and different health outcomes.
So even small variations in our environment can have an impact on our gene expression.
It seems so.
Okay.
And it's not just the broad environment like air pollution or space travel. Even the internal environment of our bodies, with all its hormones and biochemicals, can have a huge influence on gene expression.
Oh, yeah, I was wondering about that. How do hormones, for example, fit into this whole picture?
Well, hormones act as chemical messengers, traveling throughout the body and binding to specific target cells, telling them what to do. And one of the things they can do is directly influence gene expression. They can act as transcription factors, binding to DNA, and either enhancing or repressing the transcription of certain genes.
So hormones are like little commands circulating in our bloodstream, telling our genes what to do.
Exactly. And a classic example of how hormones affect gene expression is the action of estrogen in the uterus. Estrogen is essential for preparing the uterine lining for a potential pregnancy. It binds to target genes in the cells of the uterus, activating them and causing them to produce receptors for another hormone called progesterone.
So estrogen preps the uterus to be more receptive to progesterone.
Exactly. By increasing the number of progesterone receptors, estrogen makes the uterine lining more sensitive to progesterone, which is crucial for maintaining a pregnancy if fertilization occurs. It's all about timing and coordination, and hormones play a key role in orchestrating these changes in gene expression.
Wow, so it's not just about genes being turned on or off. It's about a complex interplay of hormones, transcription factors, and epigenetic tags, all working together to fine-tune the expression of our genes in response to both internal and external cues.
Exactly. It's like a beautiful, intricate dance of molecules, all working together to make life possible.
This has been an incredible deep dive into the world of gene expression. I'm so glad we had the chance to explore this together.
Me too. And for Mr. Martin's IB biology class and anyone else listening who's fascinated by biology, we hope this is giving you a deeper appreciation for how gene expression, this dynamic and highly regulated process, allows our cells to be so diverse and adaptable, even though they all share the same basic genetic blueprint.
We just scratched the surface, really. This is a vast and ever-evolving field of research with new discoveries being made all the time. So if any of this piqued your interest, we encourage you to dig deeper, explore further, and keep those questions coming.
Right. And for those of you with a strong background in biology, if anything we said sounded a bit off, or if you have a different understanding based on your own knowledge, we would love to hear from you. Feel free to leave a comment below and share your thoughts. We're always learning and growing, and we appreciate any feedback that can help us make our deep dives even more accurate and informative.
Absolutely. And if you enjoyed this deep dive, be sure to give us a like, subscribe to the show, and tell your friends. The more the merrier when it comes to exploring the fascinating world of science.
And finally, as we wrap things up, let's leave you with something to ponder. Given what we've learned about how our environment and even our parents' experiences can affect our gene expression, what do you think the future holds for our understanding of inheritance and human health? What new possibilities, what new challenges lie ahead?
That's a big question, and one that scientists are actively grappling with right now. It's an exciting time to be alive, wouldn't you say?
I couldn't agree more. The more we learn about the intricate dance of gene expression, the more we realize how much more there is to discover.
Thanks for joining us on this deep dive. Until next time, keep exploring, keep questioning, and keep those minds open.
See you all next time.